Now, as we move into chapter 8, we're getting very close now to being able to start talking about different parts of the brain and what they do. Is coming in the next chapter as we move into, we'll first be talking about the different sensory systems, and then some motor stuff as well, and how the peripheral sensory organs work, but also some of the more central regions in the brain. But before we finally get to that point, we've been covering a lot of stuff that relates to some of the more basic functional aspects of the nervous system. We have all the stuff about neurons and action potentials and synapses, and we had talked about glial cells, and we also covered some of the research methods that we will make use of to understand the function of different parts of the brain. But there's one last thing we want to cover in this general topic before we move ahead, and that is to understand the development of the brain, because this is going to help us understand some of the ways in which the brain is organized. And this occurs developmentally, and it occurs as a function both of some genetic traits. We know, of course, that we can appeal to genetics to explain certain things about the human brain, such as the fact that it looks the same across all humans, right? Everyone has a frontal lobe and a temporal lobe and a parietal and occipital lobe and brainstem and cerebellum that all look the same, do the same job more or less, right? So there's certainly some room to appeal to some genetic component to explain how the brain develops and develops its particular structural organization and functionality. But we're also going to see that experience plays a really large role. And perhaps experience plays a larger role depending on how much neocortex the brain has. So I think when we make a comparison across animal species here, and we've already covered this topic, we learned that humans have the largest encephalization quotient, but in particular, that occurs within the neocortex, right? That the main newer evolutionary part of the cortex of the brain is significantly larger in humans as a ratio to brain to body size ratio. That's the encephalization quotient. And as it turns out, it's really within the cortex that experience is important. And this concept, as I have labeled here on the title slide, is referred to as plasticity, or maybe we can call it neuroplasticity. But plasticity here refers to, just like plastic is a substance that is flexible and bendable and can be molded into different shapes. That's the concept behind neuroplasticity, is that the brain, specifically the cortex, is adaptable, right? Flexible, can be molded into different, not necessarily shapes in this case, but perhaps some various functions. And we want to understand that first developmentally, which is what this chapter is really going to focus on. But once we have the understanding of that down, we might be able to understand some issues related to the adaptability of the brain in further chapters. So right off the bat, what I want to show you here is a picture, a drawing of a human embryo. And we see the formation, the early development of the nervous system, primarily the central nervous system is what we're seeing here. We're seeing the segments of it, right? We see in red what's labeled here as the neural tube, which will eventually turn into the spinal cord. And then we see other features such as the hindbrain colored there in purple, the midbrain, and the forebrain, all right? So what we're seeing here is a view of the brain that is not quite yet fully mature, because what we know ultimately is that when we are born, the forebrain, which will include the cortex, is significantly larger than the midbrain and the hindbrain, right? This is not yet quite the case at this particular stage of embryonic development. But what is the case, you might notice, is that the head region of the embryo is quite large in comparison to the rest of the body. So the head does include the brainstem as well as the forebrain. And the head can be as much as 50% of total body length at an early stage of embryonic development. And this shows us something here. This is showing us that during prenatal development, before we are born, there's a lot of energy that goes into not just growing the entire body, of course, which is very energetically demanding, but there's a lot of energy that grows into growing our big brain, right? And that means that the head grows before the rest of the body. And human infants are markedly distinct from other animal species because of how large our heads are at birth. And so human infants are comparatively pretty helpless, because you may notice, of course, that in many other animal species, they are walking sometimes within minutes or hours after being born, or sometimes maybe within a few days, they can start walking, right? But it takes humans an entire year before they can really start walking. And even then, there's additional gains to be had over the next several years when it comes to what we might call our gait patterns, learning how to not just walk, but hop, skip, and jump, and all of that sort of thing, right? So there's really many years of not just motor development, but think of all of the other things that humans develop in childhood and how long our childhood is compared to other animal species who mature much more quickly. This process, or this difference, is referred to as neoteny. Right? Neoteny means a delayed or extended childhood. And the reason for that, at least one of the big key reasons for that, is how big our brains are, because our brains require, as I said, experience, right? The cortex in particular requires experience. And what that means is that the brain has to be sort of pre-made and ready to go by the time we're born, because there's no time to waste. You want to have the brain formed and ready to start processing all of these experiences that we will start having immediately after we are born. And that means that the rest of the body has to take its own sweet time to catch up to that. This is also referred to as a principle called cephalocaudal patterns of development. A cephalocaudal means that development proceeds in a head to tail direction, and we grow the head before we grow not just the tail. Obviously, humans don't have a tail, but just means the rest of our body, right? It goes from the top down in that way. So we're seeing here something that's happening in the early embryonic stage of development, but let's really step through some of the stages of development here. So we're going to, what I have listed right off the bat here are three prenatal stages. So at conception, when a sperm cell fertilizes an egg cell, we have a fertilized egg. That is referred to as a zygote, single cell, single fertilized egg cell. But what happens in this first period of life called the zygote is cell division, right? And all of these cells of the zygote are essentially what we'll call stem cells, right? So they're not specialized for different kinds of body tissues or anything like that. They're just cells that could develop into different body systems, but at this point, they have not yet done so. They are just multiplying, right? So there's going to be 2 weeks of cell division that occurs. And then once we start the third week, that's when we're going to start to see these cells start to specialize into different kinds of body systems. And now we call this an embryo rather than a zygote. So once we start to see those stem cells turning into actual tissues of the body, that's what the embryonic stage is defined as, is the emergence of different body systems. The first thing that happens is the formation of 3 layers of these cells. There's an inner layer called the endoderm, a middle layer called the mesoderm, and then an outer layer called the ectoderm. And each one of these 3 layers will start developing different systems. So like the endoderm will start to develop part like the digestive system and a lot of the internal organs. The ectoderm will be the skin, but it includes the nervous system. And so all of the neurons will start to form out of the ectoderm. By the end of the 8th week after conception, at this point, all of those body systems have appeared. They're clearly, of course, highly immature, but the systems are at least present. And so once everything is now actually officially present, we shift things to now calling this the fetal stage of development, which takes us from 9 weeks from conception until full term. And so primarily the description of what's happening in the fetal stage is that all of these body systems have to mature so that the child will be able to survive on its own once it's born at full term. And this, of course, includes all of the body organs, but we're going to be focusing here on the brain in particular. Something that's happening in the ectoderm of the embryo is we see the formation of something called the neural plate. And the neural plate contains something called the mantle layer. And the mantle layer is where we're going to see new neurons created, right? So this is a process called neurogenesis. We're going to talk a little bit more about that in a second here. But the neural plate folds is basically a flat, thick region of the ectoderm, the outer layer of the embryo. Something else that happens is that in the early embryonic stage, a groove starts to form in the neural plate. So it's called the neural groove, which then ultimately closes to become the neural tube. And that's what gives us the brainstem and the spinal cord, right? So in fact, coming back to that previous picture, we can see the spinal cord is forming there, what is labeled there as the neural tube, because it's essentially a hollow tube. It has the ventricle inside. But within that is where we're going to find a lot of neurons being created, this neurogenesis out of the mantle layer inside the neural tube. And so let's take a look at what happens here. So here is one viewpoint on that as well. So at the top, we're seeing 18 days after conception. So that's going to take us, just barely into that embryonic stage of life, right? And right off the bat, we're going to start seeing the neural plate forming. We see that highlighted in red here in that cross-sectional view. A few days later, it's going to start to form this groove, and it starts to fold up in the middle as it kind of folds down, and it creates this deep groove. Right in the middle, you can see that in that cross-sectional slice there, it's going to start closing, but it starts in the middle. And so the top of this, where the head would be, is not yet closed, and neither is the bottom, right? But so at this point, it's a neural groove. But by just a day later, we start to see real full closure. So we can start calling it the neural tube once it's fully closed. And again, that begins in the middle, and it just almost like a zipper, it zips up from the middle to the top where the heads would be, right? And then it also zips down to the bottom. We could talk some here about a certain developmental concerns. There are things known as neural tube defects. And what happens is that in the formation of the neural tube and closing everything up properly, sometimes there can be various reasons this does not go according to plan. Sometimes it may be genetic. Sometimes it could be the result of some sort of some external agent called a teratogen. Teratogens are things that might interfere with proper development here. Could be maybe some medication that the mom is taking that interferes with neural tube development. Examples of this include things like spina bifida, where the tail end of the spinal cord fails to close properly, but much more of a serious concern would be something called anencephaly, where the head region fails to close properly, and then the cerebral hemispheres will not develop. But if everything goes according to plan, we're going to start to see the neural tube, and here we see at 23 days, even further starting to close up, and the brain end is starting to get a little bit larger and thicker as we start to see more neurons are forming there. And then by 24 days, we're seeing at this point, the neural tube should be largely completely closed in, right? And certainly the brain is forming in there. And so this 24-day drawing right here roughly corresponds to the first picture I had up, but that was more of a see-through so that you could see inside there the different brain structures that were forming. So how does this all happen with all of these neurons being created rapidly? Well, we have to start first with the stem cells. So we're going to talk about neural stem cells. This is what we find within the neural tube. So stem cells they're called multipotential cells because of the idea that these are cells that have many different possibilities for what they might turn into, right? And one of the things that they do is, in the case of neural stem cells, is they create progenitor cells, right? So then when they create a progenitor cell, which comes from the stem cell, what's going to happen is it's going to migrate, right? And when it migrates, it's going to produce or turn into either a neuroblast, which is a very immature neuron, or it turns into a glioblast, which is an immature glial cell, right? And glial cells, of course, we learned about those like the astrocytes, the Schwann cells, oligodendrocytes, and things like that. So ultimately, the neural stem cells become progenitor cells, and then and of course, what also happens with neural stem cells is that they just sometimes just reproduce to increase their numbers, because as you should already know, we're going to be born with somewhere between 80 to 100 billion neurons in the brain. We will have nearly all of those at birth. And that means that there's a lot of cell growth that just has to happen in order to even have 80 billion of those kinds of cells, not to mention all the other cells of the body. So that's a lot of growth. Here's some steps here. So let's first talk about the cell, the creation of the cells, right? And this is called neurogenesis. In the case of having these stem cells and progenitor cells create neurons. In other cases, it's called gliogenesis if these progenitor cells turn into glial cells. Now, as I have noted here, we're going to have all of these more or less present at birth, but technically speaking, a lot of it is done by the time we're 5 months gestational age. So that means even though gestational age is closer to 9 months in length, right? The brain is really close to being, having every cell it's going to have in there, even by 5 months. What else is going on in there? There's other changes that have to happen in the brain, because just having neurons in the brain is not everything that there is to tell the whole story, which we're going to get into here. And it's also worth pointing out that if you've ever heard the myth that we do not make new neurons after a certain point of our life, and you can't replace neurons that might die, for example, that's technically not true. There is at least one core area within the brain that produces neurons for our entire life. That is the hippocampus and some regions surrounding the hippocampus. And we already know that this part of the brain plays an important role in encoding new long-term memories. And there seems to be some suggestion here then that that's probably why the hippocampus remains the active area for the rest of our lives, capable of neurogenesis, because it's quite likely that in order to be able to continue to form new long-term memories, we need neurogenesis to support that. We need new neurons to support that. There's a few things that we know about aging in the hippocampus, though, by the way. We know that it does shrink as we get older. And that shrinking of the hippocampus, one standard number that gets tossed around a lot is it loses its volume by about 1% per year. And after a certain age, like about age 50 or so, right? And so that might explain, for example, why in the aging process, we began to show some declines in memory abilities, and we have some of what we might consider to be benign age-related forgetfulness, right? That's separate from other cases of memory impairment that might result from other more significant neurodegenerative conditions, like, say, Alzheimer's disease. It seems that in aging, memory, like many other aspects of cognition, gradually declines. And this could be attributed, at least in part, to some shrinking of the hippocampus, and therefore maybe even a slowing of the neurogenesis that would naturally occur. But one of the reasons I'm mentioning that is there's also some very compelling research that shows that people who are aging and showing these declines in their size of their hippocampus also will respond pretty well to aerobic exercise interventions. And what some of these studies have reported is that after a 12-month intervention of getting people up and walking and getting their heart moving to do aerobic activity, get the heart rate into a specific target zone, that after a full year of this, they did the MRIs of their brain, structural MRIs, and saw that their hippocampus actually got larger by a factor of 2%. And that was a pretty compelling finding, because we know that it would already going to shrink by 1%. And so what they did is they didn't just hold it steady and prevent that 1% decline, but they actually gained an extra percent on top of that. So then the idea is that they were essentially not just preventing the 1 year's worth of decline, but undoing the previous year's worth of decline, right? So suggesting then that aerobic intervention helps the brain stay healthy, right? And that perhaps makes a lot of sense, but it especially makes sense with some of their additional findings where they had discovered that people who are more physically active like this have higher levels of a compound in the brain called brain-derived neurotrophic factor, or BDNF for short. And BDNF plays an important role in neurogenesis, right? There are a few of these other kind of neurotrophic factors, but they all play an important role in how the brain grows and making sure that neurogenesis can continue. And so it seems to be the case that this kind of aerobic activity not just improves overall physical health, as everyone tends to promote it for those reasons, but it promotes also increases in these compounds like BDNF that play an important role in neurogenesis, and then can, of course, improve or prevent age-related declines potentially, right? So that's where we're at for starters here. But now we have another one here, cell migration. So this is a curious thing. We see that the neurons are being created there, these progenitor cells in particular. I said they migrate. They're being created there within the neural tube. But if that's a cell that's supposed to be part of the brain, such as part of the forebrain, right, some part of the cerebral cortex, then it needs to get there, right? If it's created in the neural tube. So it actually has to move. It has to start following a pathway. So before the rest of the brain forms, we have to get some glial cells in place. So there's a specific type of glial cell called a radial glial cell. And they are called that because they radiate outward to form essentially a lattice work of pathways. And these various radial glial cells emit various chemicals that create a chemical trail. And so any particular immature neuron or progenitor cell that's migrating to a specific location, let's say it's going to be part of the prefrontal cortex, that particular neuron has to follow a unique chemical trail laid out by these glial cells that tells it how to get to the prefrontal cortex, right? And it's going to find its way there. Another part of the, other cells that are going to be part of the occipital lobe or temporal lobe or cerebellum, they have to follow different chemical trails laid out by these glia so they can get to where they're supposed to be. Right? So this migration process also requires some time, right? So it's not just about creating the neurons, but then moving them to their appropriate location. And then once they are present or in their final location, that's when they're going to start differentiating. So what do we mean here? Well, we know, for example, that we have different kinds of glial cells, right? There, like I said, there's the radial glia, there's astrocytes, Schwann cells, oligodendrocytes, and so forth. And so at this point, these immature glial cells are just very generic. They're not going to yet turn into a specific type of glial cell until they are in the right position. And the same applies to neurons, right? We have sensory neurons, motor neurons, interneurons, a few other kinds of neurons throughout the brain, right? Cerebellum has something called pyramidal neurons. There are areas in the brainstem called pyramidal tracts. So these are different shapes and types of neurons with different functions and different morphological features, right? So they start to differentiate to become these specific types of cells only once they're in place, right? And related to that is once they differentiate, they have to grow the appropriate shape, as I said, and that means dendrites and axons. So these early neurons that are just forming and migrating to the appropriate position, they don't look like neurons the way we think of them, right? With axons and dendrites. They are really just a cell body. Because all of those things branching off of it are not going to make it useful for this neuron to migrate anywhere. It's going to be a tangle of a mess, right? So we're going to wait until these neurons get to their final destination in the nervous system before we let them grow their axons and dendrites. And that process is called arborization, which means just like they're branching out like trees, right? And of course, I already mentioned, I should hope, that the word dendrite is, I think, Greek, that means tree branch. And so it really just follows that same metaphor here, that we see the formation of all of these many branches, right? And so we start growing lots and lots of these interconnections between all these neurons. And you can see that in this one little graphic at the bottom here, that even after birth, right? So you're looking at a newborn, there's just a small amount of arborization occurring between some of these neurons. But 1 month in, 3 months in, more and more branches. By 24 months, lots of these branches have been created. So once the neurons branch out, then they can start connecting to each other, right? So one neuron grows a bunch of dendrites, another neuron grows an axon. Eventually, those axon and dendrites will find each other, and then that means they can start forming synapses, right? And this is now called synaptogenesis. We have a lot of synapses in the brain. As I said, we have anywhere from 80 to 100 billion neurons in the brain. But because neurons are not just connected one to one, but rather any one neuron could have lots of other axons from other neurons coming into its dendrites. And likewise, its axon can branch off to the dendrites of many other neurons. So the total number of interconnected cells, and therefore the total number of synapses, is a huge number. And we can't exactly calculate it, but we have our best estimate here that there's 10 to the 14th power, right? So it's a lot of a lot of synapses in the brain, and that has to take a long time to develop. So one of the reasons why our brains continue to grow and get bigger, even in early childhood, it's not because we have to make more neurons, but the neurons themselves are branching out, right? That arborization process is occurring, like we just saw in the previous slide, and we're forming lots of synapses, right? This process is actually officially called exuberant synaptogenesis, just because of how rapid that is occurring. But curiously enough, we perhaps overproduce synapses, right? We make more synapses than we really need. And this gives us our next stage here, which is to actually get rid of some of them. As it turns out, there are some neurons that we might not even need, right? So we grow all these neurons, and maybe eventually we determine that some of them are unnecessary. So we're just going to let those neurons die, right? This is called apoptosis. The idea is that a neuron that's not interconnected with other neurons, and it's not really part of any functional neural network, is going to automatically kill itself, right? That's called apoptosis. But the same is true not just of individual cells, but of actual synapses as well. So as all of this arborization is happening and you get all of these synapses that are forming, some of those synapses turn out to be very useful and they're going to stick around, but other ones have no particular functionality. And so those would be essentially unnecessary, and it'd be better to just prune them away so that so we can determine and essentially the most efficient connections in the brain, because we only want to support those, because if we keep too many connections in the brain, that might be metabolically very demanding and therefore not very efficient. So we want to fine tune things, right? And that's this idea that's referred to here as neural Darwinism, is that there's this idea that just like with evolution, you got survival of the fittest, where individuals with only the right kinds of characteristics that match it or fit it to its environment will live longer and have more offspring. Same is applied here to this idea that certain synapses that are more useful for certain behaviors are going to survive, right? And they're going to get more metabolic support because they're being used, and they're going to get fed by the astrocytes that feed the neurons, right? And these other synapses will not be supported metabolically. We're going to drop off some of these cells or some of these synapses. We're going to talk a little bit more about what this might look like once we get into some of these experiments that were done by looking at the exact role of experience in determining what happens with synaptic pruning. And then lastly, our final step in all of this is the formation of the myelin sheath. And that's something we haven't talked about. We did talk about it back in the chapters on neural anatomy, that the axons are coated with myelin that speeds up nerve transmission using that process of saltatory conduction, right? But as it turns out, myelin is something that we don't want to necessarily have on axons before we've determined which synapses are useful and which ones are not. And maybe to help illustrate that, let's come back a couple of slides to this picture, right? So the axons would be the longer branches of these particular neurons. And we can see that those axons are not done growing, even if I look at the newborn or the 1-month-old. there's going to be some extra branches forming off of those axons. So that way they can connect up to a variety of other neurons in this, in here, right? And we see that perhaps right at 6 months here, we might, if you look at some of these axons, there are more branches, and therefore there are more axon terminals branching off of these axons. So what happens when you myelinate the axon is the presence of the myelin sheath actually interferes with the ability of that axon to generate new branches, right? Once you myelinate it, it's kind of done with any branching and arborization that's going to happen, right? Let's go back to where we were here. So what this means is that areas of the nervous system that do not need to really worry about the arborization process too much can start myelinating neurons or axons very early in development, prenatally. And this primarily refers to the peripheral nervous system. So the nerves of the autonomic and somatic nervous system, those generally are all fully myelinated when we are born. And so now that all that information in the peripheral nervous system is being processed pretty quickly. But as I said, it's the cortex that really requires experience. And therefore, it's in the cortex that we're going to need to determine which neurons and which synapses are the most useful and which ones are not needed. And that's going to have to take time. And we need that time to first arborize the entire brain and form all these connections, figure out which ones to keep and which ones to get rid of. And then once we're done with all of that, then we can go ahead and myelinate all of these axons within the brain, right? But we don't want to do that until that all that process is done. So that means we're not going to see myelination complete really until we're in our early childhood years, 3, 4, 5 years old. As I note here, though, there may be some additional myelination that occurs all throughout our childhood, well into our teen years, right? But I'd say a very hefty, large percentage of it is done by the time we're about 3 or 4 years old, right? This process is called now myelogenesis. That was a hard one to say, right? All right, I want to stop this video here because from here, what we're going to do for the second half of the notes is we're going to talk more about some specific research findings that inform us about exactly the role that experience plays in guiding brain development. But for now, just having this under our belts is good enough to get us going for this chapter.